Welcome to The Good Oil, the podcast where we explore the science and the stories behind DHA and its role in human health. Today's guest is Dr. Richard Bazinet, Professor Associate Chair of Research and Innovation and Canada's Research Chair in Brain Lipid Metabolism at the University of Toronto. Richard has shaped much of what we understand how the brain acquires, uses, regulates omega-3 fatty acids, especially DHA. In this first episode, we will unpack the fundamentals of DHA metabolism and dive into one of Richard's most intriguing findings, that DHA can actually inhibit its own synthesis. Welcome, Richard, for joining us. And how have you enjoyed your time in Australia so far?
This is my third time here, and third time's the charm. I'm having an absolutely great time. What a beautiful country you have here.
Excellent. It's a pleasure to have you. And knowing from my colleagues that you're at the recent conference, I'm excited about having you at this podcast. Richard, let's start with in terms of introducing yourself to the listeners, and share what drew you to this, to the actual studying of brain lipid metabolism and DHA in particular.
Sure. So, at the end of high school, we're all thinking about what we want to do in university, right? And I was interested in sports and nutrition, and was a little surprised to find out that when you go to university, you can either study sports or nutrition. They're not the same program. Yeah. So, almost a flip of a coin, I picked nutrition, started to fall in love with nutritional biochemistry, did a master's, did a PhD in lipid metabolism. And then, it was the early 2000s. And if you remember conferences, it took a while to switch the projector between speakers. There was always a hiccup, right? And so I gave a talk and I sat down, and I ended up sitting down beside who ended up being my postdoc advisor, who was studying brain metabolism. So we had a long conversation while they were switching the projectors, and I went and studied brain lipid metabolism, fell in love with it, and in 2006 I started my own program in Toronto. And almost 20 years now, we've been studying fatty acids in the brain, their role in signal transduction, and eventually how diet relates to this.
It's amazing how, destiny takes you, right?
Yeah, it doesn't take long, right, for a little twist to happen.
Not at all. Well, for the listeners who may not have a biochemistry, background, can you give a big picture view of what DHA is, but more particular of how critical it is for the brain?
Sure. So DHA is what we call an omega-3 fatty acid. It's got this long name, docosahexaenoic acid. And we'll stick to DHA, right? Yeah. But the doco means 22, and there's 22 carbons. So it's a molecule with 22 carbons. Hexa means there's 6 of these double bonds in it. And one thing that's not evident from the name of it is the position of one of those double bonds is 3 away from the end. Or 19 if you're a chemistry student, you'd name it from the other end of the molecule. But that 3 makes it important because that's the type of fat we can't make from scratch. You either have to eat it in the diet from things like fish, or you can make it from some precursors that are also omega-3 fatty acids, and that's a process that happens in the liver. The brain is really neat because the brain is almost as fat as your body fat. It's a little different, but it's pretty close. So, most of the brain is water, then half of it's fat and half of it's protein. But of that fat, about, 15% of it is DHA. And, it was discovered in the brain in 1952. And I have that paper up often on my slides in my presentation. It's in German. I can't read it, but they show the structure, which I can recognize. And, it's been 7 years we've been working on this, trying to figure out why there's so much of it in the brain and what does it do there.
Yeah. Well, let's just start with the basics. How is DHA made in the body? Could you walk us through the pathway from ALA to DHA and the key enzymes involved in that conversion process?
Yeah. So there's 2 ways. There's the shortcut is you just eat it in fish, right? And then you'd absorb it. You'd take it probably to your liver and you send it out in your blood. And eventually you'd have to get it from your blood into your brain. There's a redundancy, though, and that probably tells us something about how important it is. You can also consume this 18-carbon precursor called alpha-linolenic acid. And the naming's rough in this field, right? Because there's a linoleic acid and a linolenic. But the 18-carbon omega-3 precursor you get in most green leafy vegetables. In Canada, we're quite proud of canola oil. The Canadian oil is a source, but soybean oil is a source, flaxseed, chia seed, and a variety of others. And then you consume that, you send that 18-carbon fatty acid to your liver, and there's a series of what we call elongation and desaturation steps. The first one being a desaturation, so it's alpha-linolenic is 18 carbons with 3 double bonds. You throw another one in, then you put 2 carbons on, you throw another double bond in, more carbons, and then there's a weird step that's kind of really confusing and nobody knows why it happens, but you go a little longer than DHA. You go up to 24, you put your last set of double bonds in, and then you take 2 carbons away and you end up with DHA. And that process happens in the liver, and then that molecule is essentially DHA, and you can handle it like it was DHA. Your liver will send it out into the blood, and a variety of tissues will take it and use it as they need.
In our world, the omega-3 world, we see a lot of use of ALA in supplementation or functional foods. How efficient is this conversion in humans, from the ALA to DHA, especially if it's in infants, adults, and so forth?
Yeah, so we'll have to take a look at this in terms of stages of life. So most people will say that conversion rate of ALA to DHA is low. And one of my favorite things to do when a student says that is I say, oh, it's low. How do you know that? And they'll say, well, it's 1%. So something like 1% of the ALA gets converted to DHA. And that's low because 1% is low. It's just, if you gave me $100 and I gave you $1 back, that's a low return on investment. But there's a bigger question that it raises is how much do we need? Okay? And that becomes very, I think, age specific. So the infant has to grow a brain, especially the premature infant, right? And so the premature infant's got to take that DHA and put it into the brain, and the brain's going to grow, and they're going to keep accumulating DHA into that brain until, maybe about 2 years of age. You and I, though, our brain's not growing anymore, okay, unfortunately. But we have what's called a DHA turnover. So a little bit of a DHA gets metabolized in our brain, and a little bit gets replaced. But the assumptions are very different. So while I appreciate that everybody's like, oh, the conversion's very low, what I'm more interested in, and the bias is, well, it's low, but how much do we need? Because there could be situations where it's low and it's not enough. And I think the premature infant's a very good example of that. But in the normal healthy adult, low conversion might be enough for some things anyways. Some things anyways.
And man, there are many papers that suggest the conversion of ALA is quite limited, which I think you just sort of touched on there. How should listeners who care about their nutrition, how should they interpret that? Is dietary DHA still essential, or can the body make enough of it of its own to get those benefits?
So this is a major disagreement in the field and across countries, right? So if you come to my country, our dietary guidelines are North American, so it's Canadian-U.S. guidelines, and there's no requirement for DHA. It's not considered an essential nutrient. But if you go to some other countries, they'll say, hey, we actually have a recommendation for 250 milligrams or somewhere in that dose. So it depends where you are to some extent. And I think it's a big issue because my country, essentially when they were trying to make recommendations for DHA, threw up their hands and said, we can't do this. We give up. And determined it wasn't essential at the time. So it's a big controversy. I don't really have the answer. It's something we're really looking into, and some of our research, I think, is going to give us a hint because it's hard to do, right?
Yeah, correct.
If you give subjects a study, you randomize them to a DHA supplement, a gram a day, and you show some nice outcome, it's fantastic. That's really important. But it's hard to inform dietary guidelines with that because somebody's going to put up their hand and say, well, would 500 have worked? Would 700 have worked? And you'd have to do every study in a dose response.
Correct,
So we're a little stuck there, at least in terms of adults.
I want to just divert to one of your recent papers, the Meadow et al. 2024. this actually shocked me when I read this. It demonstrated that the DHA can suppress its own synthesis by inhibiting EPA elongation. Could you explain that to me and also to the listeners? I'm sure they'll be interested in that.
Sure. So I'll tell you what it is, and then it's got a couple implications, I think, and we're following one of them up, and one of them's already kind of in the field. So as we were talking about, there's two ways to get DHA. You either eat it or you synthesize it.
Yeah.
Well, what we found was there's a series of steps in that synthesis pathway, and eventually, it goes ALA, SDA, and then there's another omega-3 that's quite popular in fish called EPA.
Yeah.
And you can eat that in fish. And then there's one we don't talk about that much called DPA. And the step from EPA, eicosapentaenoic acid, which is a 20 carbon, there's two elongases, two different ones that can take it to docosapentaenoic, which is 22. So we found that DHA, when you consume it, inhibits those elongases and stops the EPA from going to DPA, and the EPA starts to accumulate.
Yeah.
And so that's really neat, because it's like DHA saying, hey, at some point, if you just eat a trivial amount of DHA, that doesn't happen.
Yeah.
There's a certain dose you'd have to stop your own synthesis. And we think it's really neat because it's like DHA saying, hey, there's enough of me coming in the diet. There's no longer a need to make me.
Yeah.
And that's kind of a classic biochemical feedback inhibition would be the term they would use.
Yeah.
I think it's important for a couple reasons. One is I'm a little tough on these studies. You might be getting the sense that say that the conversion rate is low to DHA. Because if you didn't control that study for your dietary DHA intake, you might, how do I word this? You might have stopped the synthesis, right?
Yeah.
And so I think we need to go back and do these studies in non-DHA consumers, maybe vegetarians or vegans, to see what their capacity is. And the hypothesis is they're going to have higher capacity than people consuming DHA.
Yeah. So in terms of the work that your team conducted, what did they observe in terms of the EPA and DHA levels when, DHA was supplemented?
Yeah, sure. So when DHA is supplemented, it almost always goes higher, okay? And that's been an important area of discussion. So people will point out that if you give ALA to humans, ALA will go up in the blood. Maybe EPA will go up a little bit, but DHA won't go up. In fact, if you give EPA to humans, EPA will go up in the blood, but DHA often won't go up in the blood. And so people point that out as like, aha, that's low synthesis. Mm-hmm. We had a study published a couple of years ago by the same author, and we were able to label the EPA. So EPA is made up of carbons. Usually carbons weigh 12. We have these fancy techniques to make them carbon 13. Mm-hmm. And we fed that to people for, I think it was 3 weeks. And we noticed that the DHA didn't go up, just like you'd expect. But when we looked at the carbon 13 content of the DHA, it was all from the EPA, right? So it had this massive amount of carbon 13 in it, and we were able to see it. So what we're interested in is called fluxes, and that sometimes things don't go up, but they can be turning over, right? And it's almost like looking at a restaurant and saying, hey, there's 100 people in this restaurant. There's only 10 people in the other restaurant. I want to own the restaurant with 100 people. But if they're sitting there all night long, you're only serving 100 people, whereas if the other one's a fast food chain, you might get 200 people going through in the evening.
Yeah.
So we're starting to look at these things from a flux perspective.
Yeah. And how confident are we with this, what you term as, feedback inhibition?
Yeah.
Are there alternative hypotheses that could explain the same data?
So what happens is people have known for a long time when you give DHA, EPA goes up. And it's a bit backwards, right? So to go back over that pathway, it usually goes ALA, EPA, and then DHA.
Correct, yeah.
But if you give DHA, EPA goes up, and there's a term for that called retroconversion. And one of my guys, Adam Metherall in the lab, was doing some studies in animals where we were giving them DHA, and we saw the EPA go up. But we have this carbon tracing technique that we use. What he noticed was that the carbon 13 content of the EPA didn't match the DHA. It was matching the ALA precursor. And that wasn't expected. We showed it again in humans, and then we spent actually a couple years working out the details of the mechanism. So we were able to get mice that lacked that elongase, and we showed that that was necessary for that EPA to rise. We were able to do weird studies where you take the ALA out of the diet, and then you can no longer get the rise in EPA. You're able to isolate the enzymes and work with them, just adding DHA and showing it inhibits them. So we put a fair amount of work showing that when you give DHA, it inhibits that elongation. There's, we've probably done about 10 studies, published 3-4 papers, showing different approaches that keep showing that finding.
And I think I heard you say that DHA converts to EPA, correct?
Yeah, so this is what they call retroconversion.
Okay.
And this gets a little tricky because DHA does convert back to EPA sometimes, but sometimes when you give DHA and you see EPA go up in the blood, people would say, aha, that's retroconversion. It's not always retroconversion.
Yeah.
Sometimes it's DHA stopping EPA metabolism, and EPA comes like a backlog and starts to accumulate. And we're interested in this because it's like DHA saying, hey, there's enough of me, and we've registered a clinical trial. We're wrapping it up, and we're just starting to present some of the results. It'll be out soon, because what we wanted to do is find the dose at which DHA says, hey, there's enough of me, stop making me. And then we're going to argue a little bit with some people that might be a marker of how much DHA you need.
So practically speaking, for someone consuming high levels of DHA, how relevant is this effect? Does it matter for nutrition or supplementation formulation?
So I'm not sure yet. There's so it matters in the literature because when people were trying to estimate DHA synthesis rates, if you've got a stop sign up, you come up with an answer that's quite low. But that's a technical issue. The other thing is we don't know how DHA works. Okay, so DHA is a fascinating molecule. It does all these things. But when people talk about, especially infants, where it can improve their cognition, it's not clear how it does that. Like I can't draw on a napkin and say it does this, and the synapse, and then you get smarter, right? And so I think what we're doing is we're just understanding the complexity of this molecule and how it even regulates kind of its sister molecules in the same pathway. And EPA is a pretty cool molecule itself because it's a precursor to a lot of anti-inflammatory things. So if you can stop EPA from going to DPA, you might have more EPA to do certain things, and it could be like an indirect effect of DHA in some of these things that we're interested physiologically. Yeah. So the short answer is we don't know, but I think it's kind of cool because it's showing how this is interacting with members of its own family.
So I want to just turn to the implications of future research, right? How might this feedback mechanism influence recommendations for dietary DHA or combined DHA, EPA, or the other way around supplementation?
So we spoke a little bit at the beginning how it's, different countries and different people, and even my colleagues, we can't agree on how much DHA we need, right? Because people make different assumptions, and it's a bit tricky to do. We've got a study registered. I won't give you the results, but I'll tell you what we're getting at. So we took a handful of vegans and we gave them a dose response. And the idea is we're going to find the first dose or the dose at which DHA says, hey, there's enough of me, stop making me. And, let's say that dose is 200 milligrams a day or whatever it ends up being. Yeah. Then we're going to say, hey, that's the body's way of telling us something, right? That we think there's enough coming into the diet, so we can slow down the synthesis, and we'll put that forward as a framework for a recommendation. Now, somebody might come along and say, "Yeah, that's fine," but it turns out that you need 600 milligrams a day to stop a heart attack. Fine. That would be more important, right? But we don't have that number yet. We don't have that kind of precision. And if somebody comes around and says, well, we did a study with 600 milligrams a day to stop heart attacks, I'm going to say, well, what about 450? Did you try 450 in your 6-year study? And they're going to say no. And I'll say, aha, so you don't know it's 450, right? And we'll continue these battles. But the idea is we'll put a number out there that's going to be a base or the start of a talking point for how much we need.
Yeah. And sort of, does genetics have a role in this? I mean, does it depend on the person's genetics in terms of the enzymes required that could have a different outcome?
Yeah, so we know there's a few things that affect DHA levels. Sex, females tend to have higher levels. One thing that's actually really important for this conversion of ALA to DHA is in that pathway, there's an omega-6 side to it. We didn't get into this, but the omega-6 side can use the same enzymes, the same elongase, the same desaturase as the ALA does. And most populations eat a lot of these omega-6s so they can clog that up and make that a bit more difficult. We're finding that there are, beyond sex, there are genetic differences in some of those individual enzymes that seem to make them faster or slower. Mm-hmm. And there's got to be a couple more things that affect this pathway. There's some things that have been shown in kind of one-off studies that I think need to be reproduced, like choline levels and some other factors. But there's going to be a handful of things that will either make the blood levels higher or the blood levels lower across time.
And suppose in your research and other research, looking ahead, what are the key questions that remain unanswered, with respect to DHA and its synthesis and its regulations and so forth?
Yeah, so I think we need to figure out how much we need. DHA is not quite like, when I went through school with something like vitamin C, if you needed 40 units, they'd say, oh, take 120, you'll be fine, right? Mm-hmm. DHA is a little ecologically sensitive right now, some of the ocean sources it comes from, and people are working on that, trying to improve fish farming productions. People can make algae. There's a lot of people working on that. But I don't think it's just one of these things we can say, we don't know how much we need, so just consume a lot and be safe. There's a whole world. There's 6 billion people, and the number's rising every time I look at it. So we need to figure that out. So I think figuring out how much DHA we need is going to be really important. Yeah. And when we figure that out, it's going to allow us to then say, that low conversion we were just talking about, it was low, and we can't get that amount. Or, hey, we can get 50% of it or maybe more. Yeah. And, how does that differ during development, and how does that differ in children, and then, adults, and then aging? And then does it differ with disease? If you have Alzheimer's disease or something, does your brain consume and destroy more DHA, so then that changes your requirement? Does that shift that? But I think that'll be the first big one to get. And then it'd be really neat if we could figure out how it worked, right? So in premature infants, I'm quite convinced it can improve their cognition. And I could give you a cheap answer and say, well, it gets into the brain, it goes into the membrane and improves their cognition. But that's just a description, right? Yeah. So it'd really be neat to figure out the mechanisms, because then, hey, maybe we could do it better with diet, but maybe somebody could find some novel approaches to treat memory disorders or improve learning in people throughout the life cycle.
Yeah. Well, in some countries now, brain disease has overtaken heart disease in terms of cause of death, right, in some countries. So what I wanted to ask you is then, can you maybe just explain, how does the DHA get to the brain as we sort of wrap up now? How does it get to the brain? Because that's really critical, right?
Yeah, so one thing I should point out is the synthesis from ALA to DHA happens predominantly in the liver. There's a couple little exceptions, okay? But the liver's the major source, ships it out, goes to most tissues. Mm-hmm. The brain does have a little capacity to make it, but the capacity within the brain, I'll give you this, it's low.
It's very low, okay.
Yeah, so it's got to come from the blood, we think, to get into the brain.
Yeah.
And then there's a lot of heated debate, because the DHA can be in a lot of different forms in the blood. And we have something called the blood-brain barrier, which creates an obstacle for getting things into the brain, okay? DHA is nice, though, because it's lipid soluble. It's a fat. And the blood-brain barrier is a membrane, and small fat-soluble molecules can cross it. Yeah. But that being said, DHA can still be in different forms in the blood. And we spent a lot of time working on this, because one idea is DHA could be in your LDL, the lipoprotein. And then that lipoprotein could go through a receptor and get into the brain. And we worked on this a lot, because there are genetic tools available where you can knock out that receptor, remove it. And that didn't work for us, and we tried 3 or 4 of those, and they didn't work. And there seemed to be 2 ways, to make a long story short, you can get DHA into the brain. One is there's what we call the free fatty acid form, or the unesterified form, or the NEFA form. And that goes into the membrane, kind of gets a little stuck there, and it seems like there's proteins that can pull it out and bring it through. The other one is DHA is what's called a lysophospholipid. So it's a type of phospholipid, but with the one DHA. And there's been a lot of confusion around that, because if I inject you with a lysophospholipid, it'll last in your blood for a long time. It sticks around, okay? If I inject you with a free fatty acid, it disappears within 2 minutes from your blood. It's just gone, right? Free fatty acid DHA disappears. But that's the stuff I injected you with. In your blood right now, so we've been talking for 15 minutes, you had a certain level of free DHA. Your level's still the same right now, because you're using it and you're replacing it, right? So we have to be very careful with some of these tracer techniques. And when you do the math, it looks like the free fatty acid form is the major source that supplies the brain. But, and I think it's an important but, a higher proportion of a lysophospholipid will get to the brain than the free fatty acid form. So it's like the brain, some people use the word prefers the lyso, that's fine, but 10 times more, I think, of your brain DHA is coming from the free form than the lyso.
Oh, thank you. That's amazing. So as we just sort of close for this edition, are there any papers or resources that you would recommend to our listeners to look for and read more about what we talked about today?
Yeah, so, if we talked largely about my work, so if you went into a website called PubMed, and you type my last name with my first initial or my two initials, RP, a lot of our papers will show up, right, including some of these newer ones. And we've got there's two things I think to look for here. There's technical papers where we do experiments, but every once in a while we write a review on the topic. Yeah. And Adam Metheralla and I wrote a review on this a couple years in Progress in Lipid Research. We're probably due for another one. But the other nice thing about PubMed is when you click on that paper, it'll say, hey, you're clicking on that paper, maybe you'd be interested in these other ones too. Okay. So I think that's a good place.
Well, we'll put up the actual website on our podcast when we film this.
And then if somebody's listening to this, you Google me and send me an email. I'd be happy to send you some papers.
Just like I did.
Yeah, exactly.
Well, Richard, thank you so much for this, and we look forward to talking to you in our next episode of The Good Oil.
Thank you.
Thanks for tuning in to The Good Oil. Today, Dr. Richard Bazinet helped us unpack how DHA is made, why dietary sources matter, and how DHA might regulate its own production. A fascinating feedback loop with real implications for nutrition. Next episode, we'll explore how DHA gets into the brain and why the journey matters. See you soon for more science behind The Good Oil.
You're listening to The Good Oil, where better health lies.